Okay, let's start. Good evening, everyone. Maybe it's good morning for somebody, and welcome to the AI Talk Season 5, Episode 1. And I'm very delighted to have you with us as we open this new phase of conversations on the evolving nature of intelligence. Today, we will have our speaker, Hokin Deng, founder of Growing AI Like a Child, which is Grow AI, which is an engineering research initiative exploring how intelligence develops and embodies cognition. And his talk, titled The Tolman Sherrington Metamorphosis of Intelligence, reveals a century-long debate between Charles Sherrington and who viewed intelligence as a hierarchy of reflexes, and Edward Tolman, who proposed an organism from cognitive maps of space and memory. So how can we trace how evolution plays invention, the hippocampus and its analog across species, enable internal models, bridging reflex and representation. Fusion links to today's AI system and ask, where are we on the evolutionary path of intelligence? The session will run for about 1 hour, which consists of a 14 minutes talk followed by 20 minutes of Q&A. Please post your questions in the chat anytime. Now, please welcome Hokin Deng for his talk, The Tolman Sherrington Metamorphosis of Intelligence. So Hokin, the floor is yours.
Yeah, thank you for your wonderful introduction. Can everyone hear me?
Yes, we can hear you.
Okay, so I think I will start sharing. Everyone should be able to see my screen.
Yes.
Okay, great. Okay, great. Okay, so I will start. So thanks for the introduction. I think today I want to work with all of you just think about intelligence in general, biological intelligence in general, and then we are going to dig in a little bit of hippocampus. Just like ask me any question anytime. Like just feel free to jump in, and then I will start. So I think I will start by first motivating every one of you like where intelligence starts in biological life. So on the left in this video, it was a work I have done with Samuel Gershman and Daniel Fur, that we were thinking about, are there learning in single cells or are there connection in single cells? So like if all of you have learned like about like simple biology, these are actually single cell organisms called paramecia. So like they are not even multi-cell yet, they are single cell yet, but you already can observe really like fine-grained like behaviors in these small organisms. And then we could see like, so we are doing some kind of Pavlovian learning here. You could see as the conditional signal, which is the shocking of the plate, and the unconditional stimulus is the electrical shock. We can already see they have some kind of like very like avoidance behaviors happening in the conditional stimulus, while quantification is required. But what I want to show you here is that even in single cells, in biological life, in biological life, even single cells already have really like intelligent behaviors. So few points here. First, like even in paramecia, all the signal molecules in the neurons are already in the place. For example, the calcium pathway, the potassium pathway, and all the PKN and PKC are already actually in paramecia, even before like the multicellular organism starts. So I would say the molecular basis of adaptive intelligence is already present before any kind of circuits of neurons like even exist. And then I will start walking through like, so we will start thinking, okay, like so now we have like multicellular organisms like over the evolutionary tree, like so what's next? So it's the sponge and placosomes, which are the next stage in our evolution, but like they don't have nervous system yet, but like so they don't have neurons, but they you do see the like diversification or specification of the function of the cells. So we do see cells that express genes that is associated with synapse. And then interestingly, like in these organisms, we have seen really like more complicated intracellular chemical signalings than what we have. So the interesting point that we have here is that like we have this multi-reusability like thesis. Basically, you can imagine there's a single cell that is really complex in their intracellular chemical signaling that is able to have all the intelligent behaviors that we have. So I think the question that I want to ask is that why we choose to form connections between neurons to have our nervous system rather than getting more and more complex in the cell, right? Like you can technically get more and more complex with all your intracellular chemical pathways, right? So I want to show you sponge that is called a sponge sneeze. So what's the sponge sneeze is that they're like these sponges are able to actually clean up the dirty stuff on their surface. So they do have this kind of intelligent reflexes, right? Like to actually clean up stuff on their surface. So I would say that they already have like intelligent reflexes, right? Like intelligent decision making. If you understand, you in some ways you can understand this as like intelligent decision making, right? And then what happened next in the evolution is called planaria and paramecia. So like they are on the tree of the so they are the next stage after sponge. So the interesting about planaria is that they already have a centralized nervous systems. So basically they have a neural cord. So they have a neural cord here, something like that, right? And then interestingly, they have two ganglia cell on the top of like like people can call that the brain. So they do have a core, they do have two ganglia here, and then they do have eyes, right? They do have the pigment cells which can detect. They do have sensory neurons already. Yeah. And the interesting thing is also the entire planaria nervous system is actually regenerative. So that like if you cut the planaria into half, they are able to grow the other half. So their entire nervous system is actually they're able to regenerate themselves. The interesting thing in human, which is that like the only thing that's not regenerative in human, which are for example, if you have a cut, like you're able to regenerate like your skin, right? So like the human nervous system is not regenerative in mammals. Like when we lost our neurons, we don't grow them back, right? Um and the other organism that's almost on the same level of the planaria is the paramecia, which if you take a neuroscience major in your school, like you probably like handle those like these organisms. So what's interesting about them? So they have they have so that's their nervous system. So you start seeing these ganglion cells, like these ganglions. So it's like a bundle of the neurons in this paramecia, right? So and they have really big neurons, like so like you start seeing this kind of like ganglion in the in the aplysia, and then they have like like you see they are quite distributed in their body, and then they do start having the emergence of the sensory neurons, which like neurons that is responsible for sensing the environment. So they do have a sensory cluster neuron and a motor cluster neuron. And then would you call the neuron like the ganglia on the top brain? Like that's a question. Like, oh, would you call these ganglion cells here? Would you call them brain? But we do see like this cluster of neurons start forming. So the pattern is that you do see like centralization of the nervous system. And then I want to introduce Eric Kandel. Eric Kandel actually won a Nobel Prize, like because of aplysia. Like so, what he found in aplysia he found the neural basis, the molecular basis of reflexes and learning of reflexes. This, Oh, okay. Like I see someone stuff. Yeah. Let me quickly walk you through this. So in the aplysia, you have the withdrawal reflex, which you touch the drill and then you withdraw, right? And then you are able to actually knock knock the tail, and then this reflex is going to become really big. So what's associative learning or Pavlovian learning, which like people probably learn about when you have a dog, right? And then like every time you ring the bell, and then you give them the food, and then they will start like they have like they start having like all the or ganglions start having all the right like so that's the associative learning or Pavlovian learning. So now we are curious like what like so the dual withdrawal is not associated with touching the siphon. And then how this association between touching the siphon and this like it become available in the neural circuits. So basically, the trick, like, so that's the neural pathway. But the trick is that like these two things needs to happen at the same time. Basically, the when you have the touch and the shock at the same time. So there's something called an adrenal cyclase is going to get synthesized, and this cyclase is going to like going to like have phosphorylate the CCAM and PKA, and this is going to go into the nucleus, which is going to activate the crab genes, and then this is going to essentially is going to form more like glutamate bubbles or glutamate vas like so basically you have all these like neurotransmitter bubbles at the presynaptic terminal. So you have more bubbles, and then basically the connection gets strengthened, right? So basically that's the nuclear like the molecular pathway between the presynaptic neuron and the postsynaptic neuron. Basically, when you activate when these two stimuli happened, there's a molecular pathway to actually getting more of the neurotransmitter bubbles, right? You get more neurotransmitter bubbles. So that's how the connection gets strengthened. So like you can read into this. So but like why is this important, and why I am mentioning to this is that everyone know like people probably like know that like there's a I, I want to emphasize when Aaron Canal figure out this is around the 80s, right? Like so that's the first paper they figure out there's a relation between the calcium and the potassium, and then like in 1981 she really find the molecular which is the molecular and neural basis of associative learning aplysia. So the classical conditioning aplysia, the molecular mechanism of this, how the connection strength is like is it becomes stronger. The molecular basis is reviewed in 1981. And then he later won Nobel Prize in this. But I want to share this because at the same time, there's someone called John Hopfield, like share the Hopfield net, which is essentially become the first computational model actually bring the Daniel Webb. So like people call this Webbian learning. It's like like Daniel Webb way before he believed that like we learn by like a connection, a cluster of neurons, and then we'll wire together, which Eric Kandel is the first person who is able to find this basis in biological organisms. And then John Hopfield have his Hopfield net, and we all know after this that David Ramaha and Jeffrey Hinton like which have a follow-up on this work like on the neural network idea how neural net like strengthen their connections. So like every time if you see a Jeffrey Hinton interview, he's on the I always know this is gonna work, right? Jeff always say that like, but like it's actually conditioned at the time. It's actually Eric Kandel and like actually find the neural basis, biological basis of how connection are strengthened in a neural net in the biology. And then all these computational neuroscientists, okay, say, oh, like that's why Jeff said, I always know that's the case. That's how we learn in the brain, right? Like he always say, like neural net is going to work. But like his belief or his conviction actually happened when he's a postdoc. Like this is the big thing that happened in neuroscience is that we do find the molecular basis and the neural basis of associative learning in neural net. Yeah, and then I think the interesting thing I want to hint on is that the first release of their backward propagation algorithm is that they mention they mention that like they think the neural net is able to learn internal representation of their environment, which is our kind of our thesis today. But they removed that later when they publish in Nature, like which is quite an interesting like thing. They do change some of the phrasing from their original UCSD technical report to their Nature paper. But like they do won a Nobel Prize later in last year. And then now I want to introduce Sir Charles Sherrington, which is so what he talked about. So Sherrington won a Nobel Prize way early before we review the molecular basis of this. Is that he basically find like the existence of reflexes like when you knock your knee, so it's called a knee reflex, right? When you knock your knee, you always like tilt your leg, right? That's found by Sherrington. So, but Sherrington, after Sherrington found the reflexes, that he have a really interesting view of intelligence, which he think the entire nervous system is a hierarchy of really complex reflexes. And then the learning is basically the changing of the strength of these reflexes. So that's Sherringtonian's view of intelligence. Like it's basically you have all these really, you can imagine you can have really complex reflexes. One reflexes is modulating the other reflexes, right? And then he believed that our nervous system is basically made up of a hierarchy of complex reflexes, and then the learning is the changing of the strength of these reflexes. Like so, that's an entire view that I presented here with Eric Kandel, Sir Sherrington. All the evidence suggests some kind of reflex intelligence is happening, right? But I think that's also the case in all the biological organisms that I've presented to you up to the time, all the way to aplysia, right? Like it's basically really complex association. But now I want to show so what's happening next in the evolution tree. So it's called a jellyfish. So they have a really diffuse neural net, but and they have really so they have all these ganglion cells. They the cluster of neurons spread around, and then you do also have the octopus. The interesting about the octopus is that they have really a decentralized nervous system, but they have a small centralized brain here. But they're big ganglion, and in their hands, like they also have really big ganglion hairs, and the ganglion here is almost at the same size of their brain. So they each of their hand is really autonomy, right? And the interesting thing is that the number of neurons that they have is actually like more than a mice. So they're actually quite intelligent. And then we have another like so what also happened next on the evolutionary tree is the insects. So there's something interesting about the insect is first, like their neuromuscular junction is completely different from us. So like they use glutamate to activate their muscle, which we use acetylcholine. Like so like they don't even share the same substrate we have in activating the muscle, and that's why we have all the insect pesticides. Basically, we actually block this, and then that is it's not harmful for humans, right? And also the interesting thing is that their neurons don't have myelins. So the myelin is a shield around your neurons that allows you to actually transfer the neural signal really far and really quick, right? So basically it's kind of like the shield. There's non conduct, so there's non-conductant, right? So they don't have myelin. In order to really have really fast speed of signal conduction, they have really giant fiber. But that can only do so much. So I would say the emergence of myelin in our neurons is actually a very important thing to our nervous system. We are really able to form really complex nervous system because we have myelin. But the insect, like unfortunately, they don't have myelin, so they are very small. Because like when you become really big, they need to make the fiber really big in order to keep the speed, right? And then even more interesting stuff is that their neurons is not spiking, like their interneurons are not the spiking neurons. So in human, we all learn about the action potential, right? So our signal is like digital, right? Like we our neuron have all the spikes, right? Their neuron don't have spikes. Their neuron actually transduct like analog signals. So insects actually have analog signals in their nervous system. It's not even like 1 or 0. It's not even spikings, right? So that's the interesting thing about like the insect nervous systems. So I will encourage, like, so what does it mean by using analog for computation instead of using like 1 and 0, like using spikes, right? So all the biological, like the octopus, the insects, and aplysia, all the biological organisms I presented here are protostomia. So how far away are are we and them, like are insects and octopus, like how far are we away from each other in the evolution? It's actually really far away. Like how far away is that when we are just a cluster of cell, like the first hole in their development become the mouth. But the first hole in our cluster of cells become the anus. So we really diverge from each other really, really far away during the evolution. It's like when we are just a cluster of cells, the first hole they form become the mouth, and the first hole that we form become the anus. So what does that mean? So at the entire nervous system of insects, octopus, aplysia, planaria, and all these animals I have presented to you just now do not share the same evolutionary trajectory to nervous system with humans. So all these are convergent evolutions. And all these are called the functional or analogs. And that's why our nervous system might look so different to them, but like they also have centralized nervous systems. So I think what I want to like leave the idea to you guys here is that when like there's much more to this, but like when we think about nervous systems in all the different animals, it's actually is the interesting thing about is that we do see convergence of design. For example, we do see that the like people start having cluster of neurons, like to have centralized nervous systems, have kind of like spinal cord in planaria, right? And then we do see the paramecia do have the spiking neuron, but the insects doesn't have the spiking neurons. So we do see conversion of design, but we also do see divergence of architectures of how they design their nervous systems, right? So I think the interesting thing is that like all of you are really great like AI scientists. It's like you guys all design different architectures, right, in your AI algorithms. Like, and then you do have conversion and divergence from some like. So I think what I want to raise here is that nature also have the same like thing. They do have a conversion of their designs, but sometimes they do have diversion of their designs. Even in the universe of biological intelligence. Yeah. So I would like I want to share this idea, like throw this idea out here, and then to for us to think about it. So what are the convergence? What are the convergence of how the intelligence should be designed, and what are the divergence? The open space that we can think about our intelligence. For example, should we do analog or should we do digital? Like the insect do the analog, but we do the digital, right? Okay, so now I will walk into chordate. So chordate is a completely, so chordate are the animals which form their first mouth, the first hole in their development into the anus. So we are descendant of chordate. So do people recognize what's this? That's actually a human infant in the embryo. So and then that's how our brain is involved, like from the beginning to the end. So like that like we have a really, so like our neurocephalon is really big, and later that become our brain. So that's what the nervous system of the human development look like. Yeah. So today I really want to like focus on the midbrain. So here, so when you think about evolution, basically that's how evolution looks like, right? I really want to share with you like here, like there's something really interesting here happening here, which is the hippocampus that we are going to talk about today, right? It's not the cerebellum and the spinal cord, which is for the movement and then for controlling the heartbeat, but it's also not the prefrontal cortex, which people think is where all the amazing stuff happening. I want to really focus on this area, which people call the midbrain. Okay, and then we're gonna watch a video on the patient HM.
1953, a desperate young man agreed to undergo an experimental procedure to remove a part of.
Can people hear the sound?
Yes, we can hear.
You can hear the sound, right? Okay.
Yeah.
His brain in an attempt to cure his seizures. Henry Molaison had begun experiencing seizures as a child following a bicycle accident where he cracked his skull. At 27 years old, his seizures were so severe and uncontrollable that he was unable to lead a normal life. His surgeon, William Scoville, was a well-known lobotomist at the time. He had spent his career performing lobotomies on psychotic patients and noted that in some of those patients, the lobotomy had the unintended consequence of relieving their seizures. And so on September 1st, 1953, William Scoville removed a thumb-sized section from both sides of Henry's brain. The majority of the medial temporal lobe was removed, including the hippocampus, amygdala, and entorhinal cortex. The surgery did alleviate some of Henry's seizures, but it also left him unable to form any new memories, eventually becoming known in the medical literature as patient HM. Our understanding of memory at the time was rudimentary. Memory was thought to be distributed throughout the entire brain and tied to other brain functions, such as perception and intellect. But the surgery didn't affect Henry's perception, nor did it dim his intellect. In fact, he scored higher on IQ tests following the surgery. Similarly, his language and motor skills were entirely preserved. It seemed the only thing he had lost was the ability to form new memories. 2 years later, in 1955, Henry Molaison was visited by a memory researcher from McGill University, Brenda Milner, who then spent the next 3 decades of her career testing and studying Henry's brain, eventually passing him on to her former student, Suzanne Corkins. Though Henry was studied extensively by both Brenda Milner and Suzanne Corkins, it was Brenda Milner's early observations that taught us most of what we know about memory. Milner found Henry to be the ideal study subject. He was highly intelligent, well-mannered, and could sit through hours and hours of testing without ever getting bored or complaining, always eager to cooperate. It was in those early years that we were able to conclude that memory, far from being distributed throughout the entire brain, was limited to specific brain structures, namely the hippocampus and adjacent perirhinal, entorhinal, and parahippocampal cortices. Together, they are now referred to as the medial temporal lobe memory system. But Henry's memory loss was an anterograde amnesia, meaning that he was unable to create any new memories while still being able to recall long-term memories from before the surgery. This is because while the medial temporal lobe is responsible for creating new memories, once these memories have been consolidated over time becoming long-term memories, they are then moved to be stored outside the medial temporal lobe in other parts of the brain. While Henry was able to recall personal events from his childhood with remarkable clarity and able to recognize the faces of famous people who were in the news before his surgery, Brenda Milner found herself having to introduce herself for the very first time every single time she met him. Milner observed that despite Henry's inability to create any new memories or remember things, he was still able to acquire new motor skills. He was able to learn new complex skills such as mirror drawing and was able to retain those skills. This was the earliest demonstration that there existed multiple memory systems that were housed in different parts of the brain. Eventually, we came to understand memory as being either declarative, the ability to recall factual information, or non-declarative, an umbrella term that describes skill learning, habit learning, and emotional memory. Things like walking, cycling, and nail biting. We now know that non-declarative memory depends on the basal ganglia, amygdala, cerebellum, and neocortex. These structures were all preserved during Henry's brain surgery in 1953, thus allowing him to acquire new skills and habits. You probably noticed that throughout this video I referred to Henry as simply Henry or as Henry Molaison, despite him being known in most of the academic literature as patient HM. There are several reasons why I insisted on this. Henry was able to provide scientists with tremendous insight on the inner workings of the human brain throughout his life. Scientists like Brenda Milner and Suzanne Corkins built their careers on the back of Henry's mind. But despite the great debt owed to him, Suzanne Corkins effectively became Henry's gatekeeper, controlling who was allowed and denied access to him. Eventually, she arranged for a distant third cousin, Thomas Mooney, to become Henry's conservator in 1992. Soon afterwards, Mooney signed a brain donation form, donating Henry's brain to Suzanne Corkins and her team. When Henry died on a cold winter afternoon in 2008, Corkins arranged to have his brain removed from his skull. She claimed ownership over his brain, his family pictures, and all of his belongings. When she saw his brain through the glass after it had been removed, she later wrote in her diary that she felt absolutely nothing at all except ecstatic. It's clear that to Corkins, Henry was not a real person. He was merely a new discovery, a scientific paper to be published, patient HM, an object to be owned and something to build your career on. Perhaps she forgot the desperate young man who trusted his life in the hands of an overly enthusiastic lobotomist.
Yeah, so patient HM is a very important subject in the history of entire neuroscience. So to just introduce so what he underwent is called lobotomy. In so it's quite infamous in a sense that so basically like lobotomy won Nobel Prize because like so what they did is they remove part of the brain is able to solve seizure, but it turns out actually when you remove the brain, like it's not actually solving the seizure because you actually remove the brain, the person become really like quiet because like their brain is removed. So and that's the reason like lobotomy is actually the reason why Nobel Prize needs to wait around like 20 or 30 years until you get because like they won the Nobel Prize like 2 years after, like I think like just like 5 to 10 years after like like this kind of surgery like is invented, and then it become a really infamous like a practice like which is people don't allow like brain removal, but it actually really hurt like brain stimulation or brain like intervention like medical medical like strategies like because of the lobotomy. There's another patient is called the severe wearing. So what patient HM have is called antegrade amnesia, which means that you cannot form new memories. So there are people actually cannot form new memories, but at the same time cannot retrieve old memory, right? So like I'm not going to like go into these videos. People can watch this like after where once I share the slide. So basically like forming new memories and retrieving old memories are both function of the hippocampus. And then like there are people who is not able to retrieve old memories and form new memories. Basically so people call him the five minutes person. So he only have a understanding of 5 minutes. He doesn't know what's happening in the past. He's also not able to like form new memory like which is coming into like his present, which is like so like they only have a 5 minute span of existence, of like understanding the existence. Yeah, so now I want to introduce a line of work is called the Illinois McQuire and Demi Hasabi's line of work of hippocampus, which they have really interesting results on what hippocampus does, not just about memory, but also a lot other stuff. So like for Illinois McQuire, like his most infamous study is the taxi driver study. Like probably some of you have heard of it. Is that when the London taxi driver have a bigger hippocampus because hippocampus is also understood to be correlated with space, right? So the taxi driver of London, like they drive a lot, so they must understand space better, and then that's why they have a bigger hippocampus. So that's a really famous study. Like people have, it's also very controversial whether it's reproducible. But his student, like Demi Hassabis, have another line of work that we are going to walk into today. So I will introduce this work for like so it's the you do see when people have hippocampus like pathology, like they are pretty bad with spatial reasoning. So that's the toss. So basically, on the pattern on the left is basically if the circle is on the right, and if that's on the left, so you have the sum, right? Otherwise, you have the raining. But the same pattern, right? On the right, there's a sum. On the left is the raining. But you can do a control experiment. If there is circle and star, then it's sum. If circle and star, it's not sum. So that's non-spatial, that's spatial. And so Demi Hassabis do show. So that's the very influential work. So Demi Hassabis do show that like the hippocampus is actually really correlated with the performance when you have hippocampus like a hippocampus deficit or hippocampus damage, you do this kind of spatial tasks like a little bit worse. So that's a very like this is very strong evidence of like the hippocampus is actually correlated with spatial reasoning in humans. And then he later have this is really like influential study in neuroscience that he showed that patient with hippocampus like with hippocampus damage is not able to imagine new experience. So what does that mean? So you can see that they have really difficulty of imagining new experience, right? So that's an example. Like so the patient is not able to really see what's happening, but the control is able to describe a thing to us, right? So that's an experiment. So what does that mean? So that means that the hippocampus is not only for retrieving and forming new memories, but at the same time it's also forming new experience, imaginations, right? So like so for Demi Hassabis, so he have these theories that like episodic memory is not really about retrieval, recall, forming new memory, but it's also about construction of the experience. So he has this theory about how you're able to construct experience from like so basically like so he have really like another theory is like so what for example, what episodic memory recall needs, like you need to understand scene construction, you need to have suggestive experience, you need to have sound, you need to have a narrative, and so on and so forth. So Demi actually have this kind of cognitive theory of how this function is linked together. Like so for yeah, yeah, and also Demi later show like it's also Demi's work, also show that the emergence of conceptual knowledge is actually highly correlated with the hippocampus, as we can see here. So basically what they did is the same task, is that they have two random subjects, two random object, and then so like people don't know like what's happening here. So basically the thing is that by giving like correct or incorrect outcome, so like the subject is able to learn these two objects is actually the same concept, the same category. So this is actually highly correlated with hippocampus. So with this line of work, Demi and McQuire really show us that like the hippocampus is not only about like retrieving and forming new memories, but it's also about forming new experience and forming new concepts, right? But like interesting thing about like Demi Hassabis as a neuroscientist, he later went on to do the AlphaGo, but he do still like most people probably know this work, and then he also like but he still have some kind of work that he want to like build artificial agents like AI, having some of his own like neuroscience work, but like that those are not the famous one. Like I think his most famous one is the AlphaFold, right? Like which I want to just mention here is that the first person which come up with the idea of predicting proteins is actually Terence Norsky. Like people probably don't know Terry, but because Terry is still doing neuroscience, like rather than AI. So he's actually a really early collaborator with Jeff. They also have the Boltzmann machines. Like they also call for the Boltzmann machine. But also want to mention here, like this person, which called the Francis Crick. So the interesting thing about Francis Crick, like is that like the early like the pioneers of the neural networks actually don't have fundings. Like because no one believed in neural networks like during that time. Like pure doing AI is all doing symbolic AI, right? but like the early donor or the early sponsor of the entire neural network or connectionist approach is actually Francis Crick. So like probably people probably all recognize this name. Francis Crick is the person who like discovered DNA, right? So but like the after path of his research career is he's actually a neuroscientist, and he's actually the sponsor of Jeff, of David Ramahan, and Terry is still at UCSD. Actually, Terry like moved away from after he have this protein work. He moved away from Johns Hopkins, and then he moved to at UCSD. Yeah, yeah. Yeah, and then like at the same time, there's another very interesting paper by Jeff and Terry Snosky is learning semantic features, and then they believe that like you're able to learn like semantics with connections, which is a complete crazy idea at that time. But now I think it's quite like obvious to everyone, like because we literally have the GPT right now. But like that's the first paper like learning some like this paper like by Jeff and Terry is actually the first paper that like they have the idea like, oh, you can actually learn language by just connections. Yeah. And then later, Demi also won a Nobel Prize with he also is the CEO of DeepMind, but he won the Nobel Prize with his AlphaGo AlphaFold work. So that's just a side story of what these people later do. But like I still want to went back to the Demi Hasabi's line of work on hippocampus. So why is that why his work of hippocampus is very important is that it really shows what memory is. Memory is a modeling system, like with his work. Is what's modeling is that forming new memories, retrieving old memories at the same time, forming new experience, which is imaginations, and forming new concepts in humans with just behavioral fMRI studies. Yeah. Like so like I want to like play back a little bit about the recent role model of the by the Genie 3, right? I think it's also like DeepMind's like Frontier and also the World Lab. But yeah, but like I'm gonna mention some of the Fei-Fei's early work on neuroscience also, and it's quite interesting how Demi and Fei-Fei have some kind of this like early work in neuroscience. But like before that, I want to like share with you a little bit about the mechanistic neural mechanisms or what's happening in hippocampus, why hippocampus is able to do all these things, right? So I'm gonna share you the work of another three Nobel last rate Nobel Prize winners on the play cells and the grease cells. So we are going to see a recording of the mice. Yeah, so what as you see already, right? So like all this color is a neuron. So we do see like there are neurons in the hippocampus literally like corresponding to the place in the real world, right? So they found the play cells, which is quite a big thing in neuroscience. So that's the idea of play cells. And also I'm gonna show you Greek cells. So that's a mouse, right? And then he's traversing around open space, and then you're going to see patterns emerging. So each red dot is a spike. Each red dot is a firing. Yeah, so what you can literally observe here is that you see you see these neurons actually having patterns like in the open space, right? Like they actually correspond to like some particular space. So we call this the grid. So these are the grid cells. So the idea is that you actually have grid-like representation of the space in the grid cells, which is in the entorhinal cortex, and the place cell is actually a downstream of the grid cells. So they are in the middle, like I think it's lateral entorhinal cortex where the place cells are. But the so like if you so like that's when they find the grid cells, and then if you do a really large scale recording of the entire population, you should be able to see terrace, right? Like that's the topology of the neural space. Yeah, and then so that's that. So the place cell and the grid cell is not the only thing. It turns out that there's a lot really interesting like other kind of cells. So you actually also have the head direction cells. So what that's what it looks like in the brain.
Looks like. So here's an animal again. We're watching where the cells fire. And every time the cell fires, we represent the animal's head and direction of pointing. So as you see, as it goes around, it drops these arrows here. And at the same time, there's a summed arrow representing the overall representation of distance. And what you can see is that most of these arrows are pointing in the north or slightly northwest direction. The cells are representing the animal's heading within the environment, irrespective of where it is in the environment. There is happier fire here as the other. These cells are found in an area just between the entorhinal cortex here and the hippocampus, and they're called head direction cells. One of the interesting things about these, it looks as though this.
Yeah, that's just John O'Keefe. Yeah, so like with here we do see the head direction cells, and the interesting thing is that like which is by James Kinnering, like so the head direction cells is actually called a allocentric head direction cell. Basically, it represents the space, the direction of the space with the allo. So it's regardless of the animals like egocentric map. So we also observe there's egocentric head direction cells, right? So like if there's open space, there's an open space, we observe that there are allocentric head direction cells in the medial entorhinal cortex, which just represent the space allocentrically in the arena. But we also found like head direction cells, which is called egocentric head direction cell, which is relative to the representation of the animals like directions. So you do have egocentric head direction cells and allocentric head direction cells at the same time in the hippocampus, and one is in the medial entorhinal cortex, the other is in the entorhinal cortex. Yeah. We also have boundary cells, which correspond to the boundary of the environment. And then so like basically they correspond to the boundary of the environment of the arena. We also have time cells. So what happened here is that basically we ask the animals to delay for a period of time, like before they go, and then only if they have the right delay, we give them reward. So we literally find in the hippocampus there are cells that correspond to, there are different cells correspond to different time in the delay. So you literally have cell that actually helps you document. Okay, like I've waited like 1 second, I've waited 2 seconds, I've waited 3 seconds. So you actually find time cells in the also in the hippocampus. And then you also find replay. Basically, when you are sleeping, your hippocampus is actually going to replay like what you encountered today. So you like basically your play cells and your grid cells is going to form a trajectory, right? And then like that's going to get replayed. So that trajectory is going to replay when you're resting. That has been found in rats. That has been found for example here in bats, and also like a lot of other, also in humans and other mammals. And the interesting thing I want to share, like there's a recent study like this year is in the birds. So there are actually gate cells in birds. So what here means that when you have a bird and then I have something like this. So when the bird went here, this neuron fire, and there's another bird went here, like the neuron fire, but it's the same when they contralaterally look at the space, the same neuron also fire. So that means that the neuron not only correspond to when the bird is physically there, it also correspond to when the bird is contralaterally looking at this place. So there are actually neurons that correspond to gaze, like gaze fields in the bird's brain. Yeah, and then interestingly, when you have contralateral, that means that it's actually egocentric, but you also have an allocentric like representation when they are physically there. Yeah, and I also want to share with you, we have mentioned evolution like just now. So I want to share with you the interesting thing about evolution is that um is hippocampus a functional monologue, a conversion of evolution throughout the entire throughout all this kind of the evolutionary tree? The truth is yes, almost all animals have some sort of hippocampus in their brain. So now I'm going to share with you with the fly brain. So insects, right? They are insects. So in the fly brain, we find the mushroom bodies, which correspond to space understanding. So like is for their space understanding. And then I'm going to show you a video.
Never underestimate the brain of a fly or the ingenuity of scientists. This fruit fly is walking on a tiny, free-rolling ball supported only by air. As the fly walks to follow a visual display, the ball moves. Virtual reality on a very small scale. The point of the apparatus is that scientists at the Genelia Research Campus in Virginia wanted to look into the fly's brain to see how it keeps track of where its body is pointed. What they found is a compass. In this composite, you can see three views simultaneously. Fly moving, the blue display the fly sees, and the brain cells, glowing green. Brain cells that are engineered to glow when they're active register the fly's movement. If fly goes right, cells on the right light up. Fly goes left, cells on the left light up. What stunned researchers is that those cells in the brain are actually arranged in a physical circle, exactly like a compass.
So the interesting thing is that in the mushroom body of the fly, like you actually have a campus, because they fly away, so you can imagine why you need a internal campus, and like the mechanisms, the cellular mechanism is actually quite well studied already. But I will just save time. I will just jump. But also it's the same mushroom body actually is corresponding to memory in the fly brain. But the problem with that, how do we even study cognitive memory in the flies? So it's pretty hard. So like the thing is that we study is actually just order memory, like to ask the sorry to ask the flies to actually just smell stuff. And the interesting thing about like octopus is that octopus also have a hippocampus in their brain, like so like their functional monologue is actually called a ventral vertical lobe, which we do see is corresponding memory and is also actually associated with their spatial learning. The interesting about about octopus is that octopus actually have allocentric representation of space, and they also have amnesia, like they actually forget stuff when they get lesion with their vertical lobe. Yeah, so they actually forget stuff. I think that's very interesting because and uh, and so in in um in, in reptiles and frogs, that's the medial palatum. So like we also find the medial palatum like in the reptiles and the frogs actually correspond to space and also memory. Like but like they only study spatial memory. It's because it's pretty hard to study like other form of memory in this kind of like animals, and it's also not very well founded. Like why did I have an extra slide? Okay, but so now I have introduced all the like the like all this like neural mechanisms that is underlying the 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 hippocampus and like and their functions like with space and memory. The interesting thing like we like that get erased is that for now on like until this point we have been only talking about physical space and physical memory, but you also can have an abstract notion of that. Basically I mean like object space or like abstract space and extra memory or extra temporal understanding, which is like narrative, right? It's not like actual time, but it's like the episodic notion of time. So so like basically Talmo, which is the another name in our title. So Talmo have this idea saying that like what we really need away from reflex intelligence is that Talmo believe that you need a cognitive map in order to have connections. You need to have latent learning. The idea is that you like so what it means is that like we remember Sherrington have the idea that our entire nervous system or our entire intelligence is just a really complex hierarchy of reflexes. What Tolman argue is that like you need to have explicit representation of the physical space and the physical memory or like abstract space or abstract memory in your representation system that is so explicit to the extent like the grid cells and the place cells in order to form a map like representations or space like basically latent representation that is impossible to get represented with reflexes or like hierarchy of reflexes. So that's the entire idea of Tolman and extending this idea is that with the same map and like map for space and like time for memory, like you can actually form like neural substrate for neural narrative and all the more abstract stuff. So like that's the action bound like which he reviewed the like in hippocampus actually like the hippocampal neuron not only respond to physical space, physical location, but also respond to the objects like the object and recent studies. So that's already really like really active research going on in the humans is that so basically like this it's pretty hard to actually study like hippocampus in humans because you cannot actually put electrode in the hippocampus of the humans, right? So what they did is that they do the MEG and then you are able to decode the theta frequency of the activation. So basically you're able to and then you do all this theta regressor and then that's you are able to also find like activation like so like grid light patterns in the representation of the hippocampus and the entorhinal cortex. So there's a very interesting study by Tim Parents like showing that like people actually respond like correspond like represent the leg length and the object profile of the bird like with the same like encoding in the hippocampus. So basically like you not only organize like the physical space, you also organize the conceptual space with hippocampus. And there's also like evidence from from rewards like so basically like in this paper they asked the humans to actually navigate navigate like in an arena with different objects, and that's the navigation trajectory of the humans. And then basically you need to form a spatial temporal like map of the space, and then people see a really strong activation in the hippocampus area. So basically you not only do you need to remember the location of the object, you also need to remember the order which you assess the object. But you see really strong activation in the hippocampus, right? Like in this space, right? But like it's very hard for us to do mechanism studies with like in humans and in concept space, right? Because like we are not allowed to put electrodes inside. And then it's the same thing for memory. So basically, like the time cells, we find the time cells like it's actually also the same thing for us to like having the episodes of narratives corresponding to the episode of the memories, and also it actually represent the goal, like where we are going after is also the same like structure. And also like in this mice like so basically where this is in mice, but like, it also show that not only do they represent the physical space but also represent the task structure, the task space. Basically the physical space is the same, but like Tim Barron's asked these asked these mice to actually traverse the maze in a order. So basically you have a task order, right? So you have a task space that you need to go to these 4 places in a specific order, right? So basically you form a task space. So what Tim Barron's found is that Tim Barron's actually found like cells in the hippocampus actually correspond to the task structure, not only the physical space, because like now the physical space is actually irrelevant. Now it's about where you are in the task, right? Like where are you in the A phase, B phase, C phase, or D phase? So it actually corresponds to the task structure, not just the physical space. So there are actually cells in the hippocampus correspond to the task structure, which is abstract. It's a abstract stuff. It's not a physical stuff. It's abstract. And also there are like cells in the hippocampus that actually depict the the movement of the mice. So let me explain what's happening here. So basically the firing, so basically you can recall a lot of like hippocampus firings, and then you are able to have a linear decoder, right? So you form a linear decoder of decoding where the mice are by basing on the hippocampus hippocampus firings. So that's the recording of place recording. But when you hold the mice, in so you don't allow the mice to move, and then you have a delay period, you actually see the same cells actually like actually have this like hypothetical firing. So it's the same firing, right? You see the same firing in the same cells, but the mice is not moving. But you see it encode a path that is going to move with the same neurons. So the hippocampus is not only just encode where you are, but also the same neurons actually like decode the path that you are going to take. Right? Like so you actually encode a path that you're going to take, right? So essentially there's an extra planning going on here. And then also there's another MEG study in humans that which like we asked the human to learn about all these object concepts, and we do see a correlation between the hippocampus activation and how we learn about the feature inference given the learning the object like object space. Yeah. And also there's a study in monkeys. Basically you are able to imagine that the reward, basically you can encode the reward magnitude and the reward probability. So given the reward magnitude and the reward probability, you are able to encode a space, right? So you like so what they asked the monkey to do is basically you can have more reward but less probability, and you can have less reward or more probability, right? But you're able to you're able to cue this with different with different picture to the monkeys, and then you ask monkey, okay, like which are you going to choose? So monkey need to put compute, right? Okay, like I'm going to get this amount of reward, but the probability is like this, right? So like which one should I choose, right? So it turns out it's also like the hippocampus is also the substrate. So you do have the amount, and the probability map is also encoded by the hippocampus. So like all these are really evidence. Okay, there's another study. So basically they asked the mice to traverse, and then the mice is going to choose going left and going right. So like we not only do we found like the hippocampus is encoding where the mice are already, like in this really long corridor, but also we are able to see like the evidence encoding. Basically, like we are able to, in the hippocampus we can find like there are neurons that actually encode the evidence of they are going to go left and going to right. So you have different cues like throughout the way, right? So basically you also see the evidence accumulation dimension that's get encoded in the mice hippocampus. So basically it's also very abstract, right? Like given a cue, like I'm going to try to decide which way I'm going to go, right? Like so you also have this like evidence dimension like in the hippocampus, like encoding like how much evidence I have accumulated. Yeah. Uh yeah, like so this study basically is that they so basically they ask the mice to go like they need to go like this, and sometimes they need to go like this, and the things that you can play with is that you can elongate the maze, you can try like you can like make the like make this like event smaller, so right, so you can actually elongate the event like or like shorten the event. So what's interesting to found here is that like so there are neurons that only represent to the event, like regardless of the time of the event. So the thing is that so when you think about event and episode, it's an abstract notion of time, right? It's not at the actual time. It's like it's not the actual how long it's going to last, but it's also the event or the episode of event. So we also find like neurons in the mice brain that is actually corresponding to the episode of the events, but the event can be longer or shorter. So it's not actual time. It's the abstract notion of time, right? That is, we also found that in the hippocampus. I think I would like, so like, are there people doing like computational neuroscience and try to build like computational models from all this what we learned from the hippocampus? Yes, there are a lot of people doing that. Like not the AI scaling people, but also like Tim Barron's have been doing that. Like some of the leading figure are, for example, Sarah Gangali at Stanford, like James Whitington in Oxford. So like there's a lot of people doing that. Sarah Gangali have a lot of work like on how hippocampus and grid-like representation is able to compute, and have a theory of what's happening in the mice brain. And also Nathaniel Doe and Perrin Pire, and you have seen Nathaniel Doe at Princeton. So there are a lot of people like doing this kind of computational neuroscience. I think I would just jump over that. But like the interesting thing, the thing about the computational neuroscience is that it's all like before deep learning. So you can see how like Saya Gangali do neuroscience is analytical mathematics. So all the models they built is analytical math model. It's not deep learning models. So there also are people. There's a recent work by Fei Li's group on cognitive map, which is like hinting on this idea. But I think like they use JSON file, like JSON for cognitive map, which is quite interesting idea. But most of the computational work is uh, is is analytical math model. Okay, so I would jump, like it seems like the time. So there's also like the ventral string and the dorsal string in the neuroscience that I will try to be quick as possible. So basically, if you have damage to ventral string, you're not able to synthesize idea. Like, but if you have damage to the dorsal string, you are not able to coordinate like your movement pretty well. So the patient DF is a patient with damage to the ventral string. So basically, he's not able not even able to tell what's in the what's in this what's in this figure. So they don't have object knowledge, but he's still able to grab stuff really well. So that's C, right? Like he's not like when they see the apple, he's not able to copy the apple, but he do remember what's an apple. So with patient R, so like I'm not go, like I'm going to jump this. So patient DF is still really good with his motor control, but patient RV, which is have the damage to the dorsal string, it doesn't have good like motor control anymore. But he's still able to have really good like he's still able to understand that's an apple, and he's able to like form the abstract representation of apple and have all the necessary feature of he's able to understand what kind of feature is making the book a book or apple an apple, right? So it's object understanding, some kind of object abstraction understanding. And the interesting is about like also here is that like patient DF is not able even to tell these two things are different, right? Like so like where that's the damage when you have the damage to ventral string. And there's a very famous Ebbinghaus illusion, like when you have the damage to ventral string, like you don't even have Ebbinghaus illusion, right? So like these two things like is actually the same size, but when you it's actually the same size, but the Ebbinghaus illusion is basically it looks smaller than this one, like to you, right? But when you have damage to the ventral string, you don't even have the Ebbinghaus illusions. But you still have but you're able still to like figure out the size pretty well, like when you're trying to do the grasping. So and then I think I'll jump this, and then there's another line of like mental disease called aphantasia, because you're not able to imagine stuff, like you don't have visual imagery. um But the interesting thing about the like image like aphantasia people, which they are not able to imagine the vivid apples, is that like so when people think about mental rotation, is that the degree you rotate is actually correspond to your reaction times. So people think like mental rotation, aphantasia people doesn't have mental rotation, but it's not true. Aphantasia people actually have mental rotation quite well. For example, like in these two tasks, basically you need to figure out if these two things are the same. And in this called the Millennium task, is that you want to figure out which hand like is holding the background. For example, like this hand is holding the background, right? And this like so that's the left hand, right? No, okay, that's the right hand that's holding the background. So like so that's the two mental rotation tasks. And then actually the aphantasia people is still able to do these two tasks quite well. So the aphantasia people, so the people without mental imagery is still able to do like mental rotation quite well. And then you also have the notion of like like motor imagery and like kinesial imagery. So like it's actually all different brain structures. So I think what I want to introduce here is a recent work by Tolman, and also we also have we are going to also release a paper on this like why this is the case. Is essentially the idea is that like rendering, so like rendering, getting a graphical representation of the of the imagery, we believe is actually separate from doing the computation over the imagery. So basically you are still able to do the computation over the imagery without actually rendering it. So they also have both of us have a lot of literatures in neuroscience and other and cognitive science saying why that's the case. So but basically like when you do the imaginations, you actually don't have the commitment to the color or the specific render details of your imagination when you think about it, right? Like so there's a lot of evidence on this, but what we want to argue is that the physical substrate of doing the computation over the imagery is actually separate from doing the rendering of the imagery. And then now I want to talk about language is a separate thing. Like I will jump. So there's a line of work by Eva Fedorenko. So the evidence they found is that when you do this like functional fMRI, you are able to separate out. So basically, whenever you do this like language task, you're able to have some kind of activation area in the brain, right? So what F really found is that you have a language network that's only for language comprehension and production, but you actually have a separate network in the brain, different network in the brain that is not for language. For example, you have a multi demand which you can actually decompose that. So, but so when you are doing the math, you're going to doing the reasoning, is actually activating a complete different area of the brain than you are doing the language, right? And also the theory of mind also have a separate new network. For example, when you're doing the language, so that's the network, but it's not the same network when you're doing the math. It's not the same network when you're doing theory of mind and other tasks. So it's separate brain areas that's getting activated in when you're doing this kind of task. So what's the point? What's the point here is that language as a function is actually separate from doing the reasoning, the function that we do the reasoning, the function we do theory of mind, and so on and so forth. But why this is important is because why this is important is because the language model is essentially all trained on language and have emergent ability of other stuff. But language itself in humans is actually a separate function than doing the reasoning and doing all the other stuff. It's not the same brain structure. Like you have a solid brain structure that's only for language. And now I want to move into Fei-Fei's early work in neuroscience. Is that okay? So what happened? So what happened when what happened like when you see a real world, when you have world perception? So in this work, Fei actually showed that you actually perceive the world earlier than you perceive language, the semantic feature of the scene. So you perceive the sensory and the feature information about the scene earlier than you from any semantic representations, right? So it's the same argument here. Is that the language is a separate thing, a separate function. Semantic understanding or semantic representation is a separate substrate in the brain than all the reasoning or world modeling or like theory of mind that we do. It's not the same thing. Like Fei, this work by Fei is also showing the same thing. Is that we perceive the world earlier than we even think about semantic representation of that world. And then he later do a fMRI study on the world perception, like perceiving the scene. And the interesting thing here is that it's literally the brain structure that we are talking about here. So it's actually called the peripheral place area, which is right next to hippocampus. So we also have a brain area. Is the PPA area is actually just for scene perception. Like it's right near the hippocampus. So like come together is that we really have a brain structure for world perception, a world like immediate world perception that's not separate from semantic world understanding. So we have a direct world understanding module in the brain that's not language at all. Okay, so how this can all come together? So I'm going to introduce. So again, is Francis Crick and Christopher Koch. Like Christopher Koch is actually also on this paper. He's actually Fei Li's PhD advisor. So so I'm going to borrow Christopher Koch's and Francis Crick framework to actually how we can we can understand all this thread of literatures together. So I would jump. So I would jump all the theory of consciousness, but I think I will walk all the way to this figure. So what essentially all this come to say, essentially become the IIT kind of the IIT theory, is that like when we are trying to understand the world, or when we are trying to perceiving the world, essentially we have this pathway that we retrieving context from the distributed representation in the brain. And what consciousness does, which is believed is the prefrontal cortex. Basically, consciousness, prefrontal cortex tells you if we are doing perception, if we are doing retrieving memory, or if we are doing imagination. Because essentially the three things are the same. Like when we think about it, we perceiving the world, we imagining the world, and we retrieving what happened, the world had happened. Is essentially all world modeling and is supplied by this pathway, which is hippocampus pathway. And hippocampus is essentially the substrate for indexing like this behavior, the indexing experience. Yeah, so basically prefrontal cortex is for monitoring and hippocampus is actually for indexing the world experience that we have. So now I want to share this theory, the little pink elephant, with Jeffrey Hinton himself introducing that.
So a lot of the problem here is that people don't know what they mean by conscious. So, for example, there's a lot of people out there who are confident that these things aren't sentient. But if you ask them, well, what do you mean by sentient? They say, I don't know, but these things aren't it. And that seems to me a fairly incoherent position. So let me take another thing that's like consciousness and sentience, and that's subjective experience. So most of us have the model that subjective experience is seeing things in an inner theater. So for example, if I drink too much, and then I tell you, I've got the subjective experience of little pink elephants floating in front of me. Most people think what that means is there's some kind of inner theater that only I can see. And in this inner theater, there's little pink elephants. And if you ask philosophers, well, what are these little pink elephants made of? They'll tell you they're made of qualia. There's pink qualia and elephant qualia and floating qualia and not that big qualia and right way up qualia, all stuck together with qualia glue. And as you can see, I don't really believe this theory. Let me give you an alternative theory of what's going on when you say, I have a subjective experience of little pink elephants floating in front of me. What's happening is I know my perceptual system's telling me fibs. I don't really believe it. It's telling me fibs. That's why I call it a subjective experience. And I want to explain to you what fibs it's trying to tell me. And the way I do that is by telling you what would have to be out there in the world for it to be telling the truth. So now I'll say the same thing as I have the subjective experience of little pink elephants floating in front of me, but without using the word subjective experience. So here we go. My perceptual system's lying to me, but if there were little pink elephants floating out there in the world, it would be telling me the truth. So you see, the funny thing about these little pink elephants is not that they're made of funny stuff and they're in an inner theater. It's that they're counterfactual, they're hypothetical. They're things that don't actually exist, but if they did exist, they'd be real elephants and they'd be colored really pink. Okay, now we can do the same with a chatbot. Suppose I train up a chatbot and it can point at things and it can see things and it can talk. And after I've trained it, I put an object in front of it and say, point at the object. And it points straight at the object. And I say, good. And then I put a prism in front of its lens. And now I put an object straight in front of it and say, point at the object, and it points off to one side. And I say, no, the object's not there. The object's actually straight in front of you, but there's a prism in front of your lens. And the chatbot says, oh, I see. The prism bent the light waves. So the object's actually straight in front of me, but I had the subjective experience that it was off to one side. And if it says that, it's using the word subjective experience exactly as we use them. So I think current multimodal chatbots can have subjective experiences, and they happen when their perceptual systems go a bit wrong. And I made this one's perceptual system go wrong by putting a prism in front of its lens.
Wow.
So I think they have subjective experiences. There isn't this magical barrier between machines and people, when people have something very special machines could never have. We have a very long history as a species of thinking we're special. We thought we were at the center of the universe. We thought we were made in the image.
Yeah, so So I will end with the conscious prior argument with from Yosha Benjo. Basically, I think what I introduced throughout here is the theory or like basically the prefrontal hippocampal like structure, which is not available in most of the mammals or like animals, but is already available in us, and maybe it's available in octopus and so on and so forth. But like this structure allows the monitoring of experience at the same time. So the prefrontal cortex does the monitoring of experience, while the hippocampus does the like indexing of experience. And this come together of us conscious experience, but at the same time is essentially how our conscious experience is basically at the same time a world modeling, a world modeling of the external world, right? Yeah. So that's all the slide that I have today. And now I'm open to questions.